In this video, I will discuss on the ribosomes and the nucleolus. So the ribosomes are actually one of the organelles that may be found in both prokaryotic and eukaryotic cells. So here in this illustration, this is obviously a prokaryotic cell. You have the nucleoid region here. And then you see the ribosomes floating about in the cytoplasm of this bacterium. In animal cells, as shown in this illustration, you can see the ribosomes, a majority of them, are attached on the rough ER. And the same is true for plant cells. So both plant cells and animal cells, the one shown a while ago, are eukaryotic cells, and the ribosomes are usually associated with the rough ER. And for all types of cells, whether prokaryotic or eukaryotic, the ribosomes are involved in protein synthesis. So, what is the structure of the ribosome? The ribosome is composed of a larger subunit, as shown here, and a smaller subunit. So these two can actually detach when the ribosome is not at work. So in prokaryotic cells, the large subunit is described to be 50S. While in eukaryotic cells, both animal and plant cells, the large subunit is described to be 60S. The small subunit in prokaryotes is described as 30S, and for that of eukaryotes, it's 40S. So, all in all, if you notice here, the combined small and large subunit of the prokaryote is 70S. Notice it is not mathematically just the addition of 50 plus 30, which is 80. So this is 70S. And for eukaryotes, the combination of the small and the large subunit is 80S instead of adding 60 plus 40 here, which is 100. So what is this S all about? Okay, S is actually sort of a unit, but it's not really a unit of measure. It stands for Svedberg. A Svedberg unit, which stands for the sedimentation rate, meaning to say the time it takes for the subunit to sediment onto the bottom of, say, a test tube. So notice that larger subunits have a higher Svedberg unit, and that is because it takes for them a shorter amount of time to actually sedimentate onto the bottom of the test tube, to sediment at the bottom of the test tube. The smaller subunits have a smaller Svedberg unit because it takes for them a longer amount of time to sediment onto the bottom of the tube. So this is quite understandable. So for example, you have sediments from a river and you put it in a test tube. If you shake the test tube and then you leave it to stand, you will notice that the larger sediments will go at the bottom first. So those are the larger subunits with the higher Svedberg units. And there will be sediments that will take really long time before going down to the bottom of the tube, and that is akin to the small subunits with the lower Svedberg units. So this is a ribbon, a three-dimensional model of the ribosomes of the ribosome of a prokaryotic cell. And here's one for a eukaryotic ribosome. So the ribosome is actually described as an enzyme in some ways. Why? Because if you can remember in genetics, the ribosome actually facilitates the translation of an mRNA into a protein. And that is a chemical reaction in a sense. And the ribosome helps in making that happen. okay, at a faster rate. So therefore, a ribosome is actually a form of an enzyme, and it's called as a ribozyme. So take note, the ribosome is actually made majorly of RNAs, okay, specifically ribosomal RNAs. And in our previous topic on nucleic acids, we noted that it's not just proteins that are enzyme-like, but also nucleic acids, particularly RNAs. And here's an example, the RNAs found in ribosomes. So how are ribosomes made? They are composed of proteins, as we see here, 1/3 protein, so the blue ones, and then 2/3 are RNA. And these are all assembled together so that you'll have a small subunit, as we see here. So this is eukaryotic ribosome, and a larger subunit as we see here. And then when these work in translation, they stick together. So particularly this RNA is the rRNA, or the ribosomal RNA. Okay, so we see here that for the small subunit, it's actually made of the 18S ribosomal RNA, and for the large subunit in eukaryotes, it's actually made of 5S rRNA plus 5.8S rRNA plus 28S rRNA. Again, do not focus on the sum of this to make the 60, okay? Because that is not how it works. Okay, remember the Svedberg unit. So, naturally, this rRNA has been transcribed from genes. Remember the mRNA being transcribed from DNA, so is the rRNA. So, the genes that actually code for the rRNA are many in one cell. So, in a single cell, there could be hundreds of these genes that would transcribe for the rRNA. Why 100 times? Because these rRNA are so essential in the cell. Remember, there are a lot of ribosomes in a cell that you need to make rRNA in bulk. Meaning many rRNA should be made at a time, not one at a time, but many at a time. And the only way to actually do that is to have many identical instructions or genes to make that rRNA. Okay, so these are transcribed simultaneously to make also around 100 rRNAs per time. Okay, so it's that fast. So all of these genes must be kept together in a single location. Otherwise, the cell, just like yourself, will lose your duplicates of your genes for the rRNA. So they need to be grouped together. And that location is together known as the rRNA gene or the rDNA. So take note, the rRNA gene is also the rDNA. And again, for the sake of organization, all of these rDNAs are gathered together in the cell in the nucleolus. So that is where the nucleolus comes in. Okay, so this one is related to your ribosomes. So this is the nucleolus, and it's a dense structure. It's a conspicuous structure in many of the eukaryotic nuclei. Okay. So here's a closer look. So you can actually have several parts labeled here. You have a fibrillar center or fibrillar centers as shown here, several fibrillar centers. You have dense fibrillar components just outside of those fibrillar centers, and then granular components for the rest of the nucleolus. So it's not bound by a membrane, but it's sort of like the pericentriolar material that is clumped together inside the nucleus. So here's a simpler illustration. So you have the fibrillar center here, the dense fibrillar component, and the granular component. So what is the composition of this fibrillar center? Okay, so remember we have 46 chromosomes. So let's talk about the human chromosomes. So in our chromosomes, of course, the rDNA, which codes for the ribosomal RNAs, may be found. But the question is, in which chromosomes? So the answer is in chromosome 13, 14, 21, and then 22. So 13, 14, 15, 21, and 22. So don't worry, you might not need to memorize that. But you see in the petite arms of these chromosomes are these red coded regions. And these are where the rDNA are found. And remember, they have to be organized together. So you have your nucleolar organizing center, or NOR. And that is exactly your fibrillar center as shown here. Okay, so that's the center there. Okay, so that's the fibrillar center. So again, the fibrillar center is made of those tips of those chromosomes, those 3 chromosomes, in humans at least, that contain the rDNA. And what about the rest of the nucleolus? Okay, so shown here in these illustrations are the rDNAs being transcribed. So you have here your rRNA gene, or your rDNA, so this long thing here. And you can see segments that are being transcribed. So this one is transcribed, then this portion is not, then this portion is transcribed again. So they are being transcribed into rRNAs. So these ones that are emerging from this long straight line are rRNAs. And then here at the tips, you can see already the starting of the formation of a ribosome. Okay, so simultaneously as the rRNAs are made, transcribed, at the tips of those, the ribosomes are already being assembled component by component. So these dense fibrillar components and the granular components are actually ribosomes that have been already started, that have begun production. So again, the genes are all kept here in the center, the rDNA, and the ribosomes that are nascent or newly formed are here in this region of the nucleolus. So that's why it appears a lot denser than the rest of the nucleus. And of course, these pre-RNAs, rRNAs, need to be modified. Number 1, through the cleaving of the whole pre-rRNA, as we see here. So this is your gene, the rDNA, and here are your rRNAs that are being made. And then there are 2 main types that are initially made. Number 1 will be the 5S rRNA, which doesn't need any modification, as we see here. And it's made using the RNA polymerase 3 enzyme. While your longer rRNA, initial rRNA here, your 45S, is made by the RNA polymerase 1 enzyme. So 5S doesn't need to be processed or cleaved, but your 45S will be. So the final products, if we skip this process, will be an 18S rRNA, a 5.8S rRNA, and a 28S rRNA. So take note, this example is for eukaryotes. Okay, so all in all, you have 5 products, 5S, 18S, 5.8S, and 28S. And they will be grouped together later on to form the small subunit and the large subunit together with the proteins to form the ribosomes. Then there is also nucleotide modification, so we will not detail so much on this anymore, just the names. There is the isomerization of uridine, and there is also the methylation of ribose. Okay, so certain RNAs actually act as enzymes for these to happen. So you have here your small nucleolar RNAs embedded in your small nucleolar RNA polypeptide. Okay, so this thing, this whole blob, and then your RNA is this ribbon thing. So they are the ones that will catalyze the reactions shown here, these 2 reactions. So here is just a detail of what's happening, of what they are doing. Okay, here we can see the formation of this loop. Here we can see the addition of a methyl group, so methylation. And then here's another look at the 45S rRNA being processed to become the 18S, 5.8S, and 28S rRNA. And then the 5S is going to be combined with the 5.8 and 28S to become the large subunit. And the 18S alone is incorporated to become the small subunit of the ribosome. Okay, so all in all, this is a summary. In the nucleus, we have the nucleolus, which organizes all of the gene to make the ribosomal RNAs. And the ribosomal RNAs are made, they are processed, they are cleaved, they are methylated, there is modification of the uridine components. And then the ribosome is assembled, okay? And once assembled, the small and the large subunits will be brought out of the nucleus via the nuclear pore complex. So you remember that. And then they are activated here in the cytoplasm, where they can then participate during translation processes. So here in the cytoplasm, the ribosomes are associated usually with a rough endoplasmic reticulum. So just a thing to remember, ribosomes have high conserved structures. Meaning to say, the ribosomes of different prokaryotes, whatever the species, are similar. And the ribosomes of eukaryotes are also similar. And that is the reason why a ribosome from a yeast cell will actually function when it is injected into a human cell. Because after all, they are very similar. They are conserved in structures. So here we just have a detail of these ribosomal RNAs, the formation of loops and the highly conserved stem. So the loops are the variable areas. So because ribosomal RNAs are very conserved, they are also the focus on the study on systematics or the classification of living things. So based primarily on the sequences found in the ribosomal DNAs. And that's also the reason why in identifying microbes, for example, you had this in your cell mall in one of your projects. It is the 16S rRNA gene that is used for the classification and identification of microbes because of their conserved qualities, as shown here in these sequences.